So an introduction to coronavirus work at the Animal Plant Health Agency. So detection, characterisation and applied research is what we undertake. The APHA is an executive agency of DEFRA in the UK, and we perform a lot of reference laboratory work. So we have international reference laboratories, national reference laboratories, and also designated labs. And that brings with it different responsibilities and different functions in relation to our competent authority reporting notifiable and reportable diseases, both within the UK, and also our responsibilities to OOE, WHO and FAO. So today I'm just going to give you a quick taste through of some of the coronavirus work that we've been involved in. And my aim as part of this network is to bring together those laboratories who already work on diagnostics and surveillance and applied R&D, and reach out to others who are trying to develop the work that they do. So we're going to cover a little bit on pan-coronaviruses, IBV from the avian world, PEDV from pigs, and also a couple of slides on our recent work with SARS-CoV-2. We engage with government in relation to new and emerging respiratory virus threats advisory group, and we bring our expertise from other respiratory viruses to this grouping. And we also liaise with our public health colleagues from our veterinary perspective via the human-animal infections and risk surveillance group. So we are inputting to those coordinators to provide the government with the information that it needs. So from a pan coronavirus perspective, ACBAR uses a microarray approach, which is able to detect a wide range of coronaviruses from the different lineages. And that is looking at a range of viruses from different species. And this includes wildlife, livestock, and also potentially onto humans for characterization. Next slide, please. So in terms of the avian infectious bronchitis virus, we have diagnostic tools which are mainly focused around PCR. And the key to being able to deliver these from a science, surveillance and diagnostic perspective is actually that having the quality assurance and validation and accreditation of your tests, which is something that some of our groups specialise in at APHA. And we work with collaborators across our European labs to actually run these proficiency training panels and prepare quality assurance documentation in order to be able to deliver these. And we are looking to be able to assist others in these areas in order to refine the quality of their diagnostic and surveillance. Next slide, please. In terms of applied R&D, we are constantly improving and making sure that our tests are fit for purpose and that they are able to capture new and emerging viral variants that come across in terms of coronaviruses, but also many other families of viruses. So Scott and his team have been keeping contact with those in the Netherlands and Poland, where we're seeing emerging IBV strains, in order to ensure that the detection ability is kept up to date. Next slide, please. So Falco and Jean-Pierre focus on swine virology, and in terms of coronaviruses, they've done some investigations around pig endemic diarrhea virus, which hasn't occurred in the UK for some time, but we have that surveillance capability, detection and diagnosis as part of our national reference laboratory function. Next slide, please. So that includes doing a considerable amount of genome analyses and actually comparing those to other isolates from around the globe. You can see that this isolate from England was related to some of the more historic viruses, but had also begun to deviate away. There has been no PEDV detected in the UK since this particular event back in 1999 and 2000, but viruses have continued to evolve in the PED world across Europe, USA and Canada and China, and still in America, they are suffering huge economic losses from this particular production disease. Next slide, please. So a little bit of information then about me and what we are most recently bringing to the consortia. One of the specialties that we have is around animal models of infectious diseases, from swine infection models where we look at pigs alone, wild boar, but also transmission from pigs to poultry, for example. We have the avian infectious disease models where we can look at both poultry, game birds, and wild birds such as pigeons. And from a One Health perspective, we have developed some innovative transmission models where we have cross-species infections, which might be from pigs to ferrets. Both pigs and ferrets can act as a model of human disease, so they're useful in both those capacities. But we've also done work looking at transmission from ferrets to birds, and most recently been looking at the SARS-CoV-2 viruses in the ferret model, which is a mild to asymptomatic model for humans, but also we can ask questions about the mink outbreaks that have been occurring across the world. So in terms of the applied R&D, I've just listed a selection of projects down there with funding from various sources. And we're hoping to bring in this connectivity with these other projects into the UK ICN network. So creating a network of networks and reaching out to members across the globe. Next slide, please. So we have a pathology specialist, and they've been applying their tools to looking at both SARS-1 and SARS-CoV-2, and also MERS from a protein detection and also a genome detection level. You can see some experimental results from our animal models there, looking at virus shedding, also survival of virus on pelts, for example, and that work was carried out by Helen Everett in our team. Next slide, please. We also have been taking a look at the ACE2 receptors for SARS-CoV-2 and comparing them across a wide range of species and looking at sequence changes in the coronavirus spike protein and how that interacts with the different species ACE2 receptors. Next slide, please. So in terms of diagnosis, in pets, we have been working with DEFRA as our competent authority and then reporting this onto the OAE, where we have detection of SARS-CoV-2 in animals. So you can see that we had a couple of investigations in 2020 and we've had 6 in 2021. We've had 2 SARS-CoV cases confirmed following private veterinary test investigations. So this is part of us acting on behalf of the competent authority and then confirming those with our accredited and validated tests. So we've had 1 positive in a cat and the 2nd positive in a dog. Next slide, please. So other aspects that I haven't been able to cover this morning, looking at the risks of SARS-CoV-2 in wildlife. We have the National Wildlife Management Centre at APHA, and we've done some investigations around the risk of a wide range of species of wildlife. And this is in respect to potential reverse zoonoses and reservoir establishments from COVID infection in humans and passing it back into the animal world. We also have a strong team of epidemiologists, biomathematicians and risk researchers who look at all things in relation to risk, which might include public health concerns in terms of international travel, but also around wildlife and livestock.